Hi everyone! Today we're going to be talking about Chapter 4, which is all about the actual way that we transmit genetic information from one generation to the next. So we're going to talk about what's called single gene inheritance. And this is Chapter 4 in our textbook. And I'm actually going to break this apart into two different sections. So in this first lecture, we're going to talk about the basics of single gene inheritance. So the idea, one gene, one trait. And we're going to talk through that first. And then in our second lecture, we're going to look at how we use a tool called a pedigree to follow traits through different generations. So in this chapter, we're going to sort of talk about how we discovered how traits are passed from one generation to the next. And it's largely in part due to the work of Gregor Mendel. So we'll talk about his experiments. And then we're going to talk about some of the important things that he discovered. So we're going to talk about the law of segregation and independent assortment. And we're going to talk about dominant traits and recessive traits, and there's a lot of vocabulary and terminology in this chapter, so pay particular attention to these terms. So let's talk a little bit about Mr. Mendel. He was a 19th century monk, but in addition to being a monk, he was also a mathematician, and he had a profound interest in understanding how traits were inherited. And so he knew there was some math behind it, and his mathematical mind wanted to figure out how we could see traits being passed on from one generation to the next and actually describe it. So it took a long time for his work to actually be recognized. He did his experiments, which we'll talk through, and he sort of patterned out these ideas on how traits are passed on before we knew anything about DNA, before we knew anything about genes or anything like that. And he had all this information, and he presented it sort of very locally. He presented it to other Franciscan monks and published it in very small publications, but nobody really understood what he had come up with and the importance and significance of it. And it was more than 50 years later, when other scientists were doing the same types of experiments, that they stumbled upon it and they said, wow, he did it first. He actually understood this long before any of us did. So in addition to being a mathematician, Mr. Mendel was also very fond of plants. He was a bit of a botanist, so he enjoyed what we call sort of as a hobby, botany as a hobby. So he liked plants, and he was interested in plants. And so for his experiments, he chose pea plants. And pea plants are important because pea plants had a variety of traits that had two distinct forms, and they were very easy to describe. So we looked at lots of traits related to the seed and the seed pods. So we looked at the form, the color of the pods, the form of the pods, and then other things like where the flowers were, how tall the plants were, and what the seed coat color was. So these 7 traits were what he used to try to determine how things are passed from one generation to the next. So what's the pattern of inheritance is what we're going to call this. So what Mendel was studying was what we're referring to as single gene traits. So one gene, one trait. So what you'll find is, as we continue on in the course, is that a large variety of our traits, especially our human traits, are much more complicated than single gene, single trait. But for simplicity right now, let's just talk through this idea of one gene, one trait. And what also made it very simple is that these traits, there were only 2 forms or alleles. So if you remember, an allele is a form of a trait. So in the case of the table that we just saw, so here let's go back up to that real quick. In the case of this table that we just saw, these are different alleles. So for seed form, you have a round allele and a wrinkled allele. So 2 different forms of this gene. You have a yellow allele and a green allele. So alleles are just alternate forms of a gene. So they are a different phenotype, right? So you can be yellow or green for your seed color. You can have wrinkled or round seeds for your seed form. Let's get into some vocabulary that we're going to be using now throughout the rest of this lecture. I want to talk about genotypes, and I want to talk about phenotypes. So your genotype is the combination of alleles that you have, okay? And we use letters to represent alleles. Big capital letters and lowercase letters, which I'll explain why we use those differently in a moment. But your genotype is basically a combination of letters, and those letters represent what genes you have, what form of genes you have. And if you have two of the same allele, if your alleles are identical to each other, we say that you are homozygous for the trait. If you have two different alleles, we say you are heterozygous for this trait. So homo meaning same, hetero meaning different. That's a good way to keep that straight. So homozygous, you have two of the same. Heterozygous, you have two different. Your genotype directly impacts what your phenotype is. So your phenotype is the physical outcome of that genotype. So if you happen to be a pea plant that has two big capital T alleles, which means that each T allele actually stands for being a tall plant, and you have two of those alleles, your genotype would be big T, but your phenotype, what you look like, would happen to be tall. So let me give you some examples here. I'm going to come back to dominant and recessive in just a moment. So I want to get into alleles for a moment. So again, we use a combination of letters to describe the genotype that you have. So for dominant alleles, we have a capital letter. So a dominant allele is a trait that will show up in an individual even if you only have one of those alleles. Okay, so for example, in pea plants, being a tall pea plant is a dominant trait. So we use a capital letter T to mean tall. If you aren't tall, if you are a short pea plant, then you don't have that capital T allele. You have a lowercase t allele instead. So we use a little shorthand with letters. Capital means a dominant allele, and lowercase means a recessive allele. So what does a recessive allele mean? A recessive allele is one where you have to have two copies in order for the trait to be observed. So in order to be a short pea plant, you have to have two lowercase t's. If you have even one uppercase T, capital T, you will be a tall pea plant. Okay? So then the question is, how do we know what traits are dominant and what traits are recessive? Well, it all starts off with creating pure breed individuals. So what's a pure breed individual? A pure breed individual is an individual who will produce offspring that always look like them, right? So if you take two pure breeding or true breeding individuals and you cross them together, in the case of pea plants, we're going to cross them together, all of the pea plants will come out looking exactly like the parents for that particular trait. So for example, if we had two pea plants that have white flowers and we crossed them together, all of their offspring would only produce white flowers. So we would say that these plants are true breeding for white flowers. That means that they're going to have two of the same allele, right? So they have two of the same allele. So in this case, we're creating homozygous individuals, individuals that have two of the same alleles for a trait. So in this case, two white flowered parents will make white flowered offspring. So again, the idea we could use it in any way. We could take two short parents and all of the offspring would be short. We could take two tall parent pea plants and all of the offspring would be tall. Again, the idea here is that we're creating homozygous individuals. So both of your alleles for that trait are identical. Okay? Now, as it turns out, being short is recessive and being tall is dominant. So in this case, we've got two recessive alleles for short pea plants or two dominant alleles for tall pea plants. But regardless, we have individuals who are true breeding for that trait. All right, so Mendel created a bunch of true breeds for all of those traits that we looked at earlier. So there were 7 different genes. There are 14 different combinations here that we can look at as far as true breeding individuals. So his next step was to crossbreed them. So he took individuals that were true bred for different versions of the trait. So if we took those tall, let's do the flowers because that's what's used in example here. If we took those white flowered individuals and we crossed them with purple flowered individuals, as it turns out, we get offspring that have all purple flowers. So what that tells us is that even though we've got 2 alleles here and 2 alleles here, purple is dominant to white. So because you'll inherit 1 copy of the allele from this parent and 1 copy of the allele from the white parent, that would give you 1 purple allele and 1 white allele. The white allele is recessive. It cannot be seen because it's being masked by the purple allele. We could do the same thing with tall and short pea plants. So if you cross a tall parent pea plant with a short parent pea plant, all of the offspring are tall. So this now creates different individuals. Rather than the homozygous individuals we started with, now we have heterozygous individuals, individuals who have 1 of each allele. They got 1 short allele from one parent and 1 tall allele from the other parent. So now we're creating heterozygotes. This tells us, again, which trait is dominant and which trait is recessive, because the dominant trait is the one that will always show up because you only need 1 copy of that allele. So after he did this, after he created his true breeds and he created all of his crossbreeds, Mendel came up with the first of sort of our rules of inheritance. And our first rule of inheritance is called the law of segregation. And what the law of segregation says is that alleles are distributed into separate gametes during meiosis. And again, we know this because we've been through the process of meiosis. We know that you get 1 chromosome from mom and 1 chromosome from dad. So again, we kind of already covered this, but again, this was revolutionary when Mendel came up with this idea. At the time, he didn't know what they were. He didn't know they were chromosomes. He didn't know they were alleles. He called them units, and that you inherited 1 from each parent. And again, we have 1 allele for each trait is what we sort of summarize from this law of segregation. And what's important here is that the allele that is not expressed, so the recessive allele, it doesn't disappear. It doesn't go away. It's there, but that trait is not being expressed because the dominant trait is masking it. So again, in meiosis, we've seen this, right? So during the process of meiosis, here are our chromosomes. They're replicated, and then after 2 meiotic divisions, we end up with 1 of each chromosome in each of these gametes. And then again, in the process of fertilization, the gametes will combine. So this isn't new necessarily for us because we saw this in meiosis, but this is a new way to describe what Mendel was seeing with the pea plants. So we have a handy little tool in order to help us visualize how traits separate into gametes and what possible gametic combinations we can get. And it's called a Punnett square. And a Punnett square is a predictive tool. We can use this as a way to predict offspring phenotypes if we know the parent genotypes. So all we need to know is something about the parents. Are they homozygous or heterozygous for a trait, dominant or recessive for that trait? And with that information, we can predict the likelihood of what their offspring is going to look like. So let's start off with a parent who is homozygous for a trait. And in this case, they are homozygous dominant. Okay, they have 2 capital letters. The trait is the A trait. So they're only going to be able to produce gametes with all A's, right? A parent who is heterozygous for a trait, however, has 2 possible gametes. They can produce a gamete who either has the dominant allele, the capital letter, or the recessive allele, the lowercase letter. All right, so what we do is we use a 4 square. So a Punnett square is a 4 square, and we're going to put the possible gametes on the top and side of the square, and then we're going to combine them in the center boxes. So let me show you what this looks like. Okay, so here's our Punnett square. Let's go back to those tall pea plants again. Let's say we take 2 parents who are both tall, homozygous tall, they're true breeding for that trait tall. So parent number 1, again, this is their possible alleles. They can only make the big T allele. And parent number 2 is also true breeding, so their alleles go here on the left. Okay, then what we're going to do is we're simply going to combine these two alleles into these center boxes. So we go to the top and the left right here, and then we go top and the bottom left and combine them, and then we go over here to the top right, and again we combine the letters together, and this is what we get. We get individuals who are all homozygous dominant. So a little bit of notation here, P1 simply means the parent generation. Genotype, again, is the combination of letters that they have, and phenotype is how they look. They're tall. So these two parents, if they crossed, we would have all homozygous tall pea plants. And then the other notation we use is F1, which just denotes the first generation, so that means offspring. All right, so what happens if we do a cross here? Let's take a homozygous tall pea plant and cross it with a homozygous short pea plant. Same thing, we're going to combine all the letters together, again, making sure that we're using the top and the bottom squares on all of these. And what we get is individuals who are all heterozygous for a trait, right? They all are tall, that's their phenotype, but their genotype is heterozygous. They got one of each allele from each parent. Okay, so the next cross we're going to do is what's called a monohybrid cross, and this is a very popular cross where we're going to actually take those heterozygous individuals and we're going to cross them together. And now you're going to see that we've got a few options here. Rather than all of the offspring looking the same, now we have some possibilities for some different offspring. So in this case, once again, our two tall alleles combine to create a homozygous tall individual, and then our big T allele and our little t allele combine to create a heterozygous individual. Over here we also have another heterozygous individual, and our last individual is different. Our last individual is homozygous recessive, short. So now we have something a little bit different. When we're looking at our predicted offspring, we have individuals who are 3 tall individuals to 1 short individual, or you could also look at it as 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. So these are ratios that we use then to sort of describe our offspring. Okay, let's do a little comprehension check and take as much time as you need with this question. But in pea plants, purple flowers are dominant to white flowers. If 2 heterozygous purple-flowered plants are crossed with one another, what proportion of the offspring will also have purple flowers? None, 1/2, 1/4, or 3/4? So I'll give you the answer, but if you want to pause and draw out your Punnett square and figure it out, I highly recommend it. But the answer is 3/4, right? So you will have 2 heterozygote individuals and 1 homozygous dominant individual. And if you want to look at that math again, go back to my previous example, and you can see exactly how that worked out for the tall pea plants. So the last thing I'm going to really briefly mention, and I'm going to go through this kind of quickly because this is it's important, but again, it's I want you more to focus on using Punnett squares for single gene traits. Mendel next did 2 genes at a time. So after he had done all of his monohybrid crosses, he started to do dihybrid crosses where he looked at 2 different traits at the same time to see what would happen. And we can use Punnett squares in some cases for dihybrid crosses. Again, don't get too caught up in this Punnett square right now. I'm just illustrating that you can do it. But basically what came out of his dihybrid cross work was the fact that we have something called the law of independent assortment. And what it says is that 2 genes on different chromosomes will segregate their alleles independently. So what this means is the inheritance of an allele of one gene has no influence on the inheritance of a second gene. So you could be tall and have purple flowers, or you could be tall and have white flowers, or you could be short and have purple flowers. So traits don't follow each other. That's basically what Mendel was saying, is that traits can be inherited separately, and that's the law of independent assortment. So these last couple slides are about dihybrid crosses, but I'm going to kind of skip over this because, to be honest with you, it's too hard and it's too difficult. We can use probability to predict dihybrid crosses, but even that, I'm just going to kind of skip over this, to be honest with you. If you're super interested, go ahead and refer to your textbook when it comes to probability and dihybrid crosses. But we're just going to focus on monohybrid crosses, so I'm going to skip that. So let's do our last comprehension check here. 2 genes that are located on different chromosomes will blank during meiosis. Segregate randomly, assort independently, not obey Mendel's laws, or not be transmitted. And the answer is they'll assort independently. So that's the law of independent assortment. So again, traits don't follow each other. You can have a large combination of traits, and that's where we get all of the wonderful diversity we see in living things. Okay, important terms and important concepts. I will have practice genetic problems to practice these Punnett squares. They're very easy to do, but they just take a little practice, and I will have practice problems posted on Blackboard for those.